Welcome. I am Raoul Concepcion, the Chief Science Officer at U.S. Urology Partners based in Nashville, Tennessee. It's a pleasure to deliver this lecture today, which I am entitling Genomic Testing in Prostate Cancer and the Role of the Urologist. These are my disclosures. This slide really represents the seminal paper that was published in 2016 that really started our interest in looking at the hereditary component of patients with metastatic prostate cancer. This was published by Colin Pritchard, and what they did through 7 institutions, they recruited close to 700 patients that were diagnosed with metastatic prostate cancer and conducted germline testing on these close to 700 patients. And these patients were not pre-screened for family history, age, ethnicity, so it truly was a representation of patients with metastatic disease, again, across 7 sites. And the key point with regards to this paper is really listed on bullet point number 4, where of these close to 700 patients, about 84 or 12% of these patients actually were identified with germline mutations. So this really started the whole interest for the urology world to be more focused on the hereditary component. And what's particularly interesting about this is that relative to patients with localized disease, the incidence for localized disease was about 4.6%. And this was from an established database. And so the upshot of this is that patients with metastatic prostate cancer have about a 2.5-fold increase incidence versus those with localized cancer of germline mutations. Now, many of you have seen this alphabet soup, if you will, of the genes that have been commonly associated. And what I'd like to do really is to try to simplify this for the urologists who are not necessarily that well versed in this whole lexicon of genetics and try to organize your thoughts on how to best think about this. So most of these genes that you see listed or that you're familiar with or have heard about really can be classified as DNA damage response genes. So you will see this abbreviation DDR. So again, the genes that we're going to discuss and the importance of testing really all come down to DNA damage repair. So many of you are familiar with PARP. PARP is a family of about 17 genes, and really they are involved in post-translational modification and really with identification and excision of damaged DNA. Then you have genes that are associated with repair. And the easiest way to think about this is that there are genes that are involved in single-stranded break repair, and those really are down to mismatch repair, or MMR. And these are the genes that, again, that you will commonly see: MSH2, MSH6, MLH1, and PMS2. And to put that into context, and the importance of those genes is that those are the ones that are also associated with Lynch syndrome, which is a hereditary colorectal cancer, but also has some GU implications. You also have genes that are important in double-stranded break repair, and those are the ones that we classify as homologous recombination, or HRR, and those are the ones that, again, you commonly will see as BRCA1, BRCA2, ATM, and PALB2. So again, this alphabet soup of genes that you will see that are associated with prostate cancer really fall under DNA damage, and you have really 2 big categories of mismatch repair and homologous recombination. And then when you get really unregulated cell growth, that is really the definition of cancer. So some definitions that I think we all need to be familiar with are things that you already heard and already know. So there are genetics, And this really revolves around the study of genes and their inherited characteristics, which was discovered back in the 19th century by the monk Gregor Mendel. The genome is the organism's complete DNA set. You have exons, which are really the coding areas of the genes on the DNA. And surprisingly, that only accounts for about 2% of your entire DNA. And when they started the Human Genome Project and finished it, it was really the sequencing of the exons. However, you also have introns, which are these areas in between the exons that are non-coding areas, and that's about 25% of the DNA. So you have a vast majority of your DNA that we really call junk DNA, and that goes back really for almost centuries, as these are areas where viruses can be housed and these types of things. But again, in terms of our understanding relative to the implications of what the Human Genome Project was able to accomplish, is really the exons, which again is just a very small minority of your DNA. Genomics is the complex analysis of the patient's genes, their interactions with other genes, as well as the environment that may result in unregulated cell growth, which again is by definition cancer. And we're also starting to understand the importance of these non-coding areas, these introns, on how they will affect the coding and the ultimate manifestation or expressions of proteins. So it's critical as we learn about genomic testing, genetic testing, however you want to think about this, there are some testing basics. And we talk about germline testing or inherited testing versus somatic. And so it's very important when you start to get into ordering genomic testing and incorporating that into your practice, you have a basic understanding of the difference. So germline or inherited testing are changes or mutations in DNA that are present in the reproductive cells of the patient. So again, this is the DNA that you inherit from your parents. Obviously, whether it's from your father, it's going to be in the sperm. Whether it's from your mother, it's going to be in the ovum, and it's going to be passed from generation to generation. So that is the definition of germline testing. So if you have a variant or a mutation that is passed down from your parents, it's going to be in every cell of the body that has a nucleus. So it can be in saliva, which basically will get shedded cells from the inside of your mouth. A lot of testing is done on white blood cells, but the point is you have to have a nucleated cell. So it obviously cannot be a red blood cell or a platelet. Somatic testing, when you hear that terminology, is basically mutations that can be defined as any alteration in DNA that occurs after conception. So that is tumor testing. So when we talk about somatic testing, what the labs are doing is taking a sample of the tumor and performing the sequencing on that particular cell. That will not be identified in every cell of the body. So it's very important that you understand when you start looking at results, start ordering testing, that it's important to understand the difference of whether or not you're going to order germline or somatic testing. As we all know, your genes are housed in your DNA, which is then rolled into a double helix. They're basically on your chromosomes inside the nucleus of a human cell. We have 22 chromosomes as well as a pair of sex chromosomes. And the chromosomes are really, again, this is very basic. They're organized with contributions from both your mother and father, where you have basically a P arm or a petite arm, a short arm, and a Q arm or a long arm. And so again, what you inherit is really housed in your chromatids. So when we start naming the location of the chromosomes, we actually have to identify them by the locus, which is the position occupied by a gene on a chromosome. And again, you have 2 copies of each, which is an allele. So that's an alternative version of a gene on that specific locus. So when you see chromosomes, when you see genes that are labeled, each chromosome position, each gene position is located on a band. It's given a number, and it's the combination of the band number and arm which gives you the locus. For example, the BRCA1 gene is on chromosome 17. It originates on the Q arm at position 21. BRCA2, when you see that listed, is located on chromosome 13 on the Q arm at position 23. So that is why it's important to understand, if you're interested in trying to read these reports, which you should be, why these are identified as such. And it's very standardized nomenclature. And really what you have to remember is that, again, these are concepts that we've learned back to our basic days of biology, which is really central dogma. And the central dogma is that you have DNA, which then transcribes into RNA, and then the RNA then translates into a protein. So that is really shown here, where again, you have DNA that then unwinds into an RNA, and then finally translation is the coding of the protein that's being expressed. So even though we talk about genes, it's important to understand that genes ultimately will result in a protein. And it's these proteins that are actually involved in the DNA repair. And if you have a mutation or a variant that alters the function of those proteins, that is where the problems arise. So it's also important to remember that in the genetic code, each amino acid is coded for by particular base pairs. And in DNA, remember, these are all nucleotides, and so you have purines and pyrimidines. And in RNA, you also have that same type of pairing. So what tends to happen, and again, this is very organized if you look at this slide, once you get to messenger RNA, which again translates into a protein, the body knows that it has to start the translation at a certain codon, which is for methionine or AUG. Then you have multiple sequences that can code for the 20 amino acids that we know build polypeptides or proteins. The body knows when to stop the translation as we have these stop codons. So again, even it's important to recognize that the DNA transcribes into RNA, and it is the translation of the RNA which results into protein. And why I'm spending a lot of time on this, it is because of the fact that is these alterations in these nucleotides that are the actual mutations or variants. So remember, proteins are made up of 20 amino acids, and the amino acids, even though there's only 20 of them, we have what's called this degenerate code, is that you can have, for each amino acid, you can have sequences that result. So even though there are only 20 amino acids, each amino acid has the ability to be produced by multiple codes. And again, why that is important is because it is these alterations, these mutations that happen within these exons, these codons, that can result in a protein change. So again, the start codon is automatically AUG, which is methionine, but the stop codons can be really coded for by 3 distinct sequences, UAA, UGA, UAG. And so to put this into perspective, what happens is that if you have a change in those nucleotides, a variation, and it results in one of those stop codons, you can stop the translation from the RNA into the protein can stop, and this is where you run into the problem. So what's happening now is that we have a very deep understanding. We have the technology to really sequence all these variants, all these mutations, all these nucleotide sequences. But our understanding of how this relates to actual clinical experience, cancer genetics, really outpaces our knowledge. So I actually stole this slide from Colin Pritchard at the University of Washington, and it's akin to the young man on the bottom right of your screen. He just wants to get his tricycle down to his friend down the street. And he can certainly get there by riding it. However, he's being pulled by this 747. So our clinical knowledge, our appreciation of the implications of genetics is really akin to the young man riding a tricycle. Our knowledge is pretty limited. However, we're driving this with this next generation sequencing, and we're generating a tremendous amount of data, which we are trying to grasp and we're trying to interpret. So I think this is a very appropriate quote from 1931. In every case of every malady, there are two sets of factors at work in the formation of the morbid picture, mainly internal or constitutional factors inherent in the sufferer and usually inherited from his forebears, and external ones. So in this case, the malady is cancer. And what's important in the development of cancer is this interplay, the genomics, the relation of the constitutional factors vis-a-vis the genes which you inherit from your parents, and the interaction with the environment. So and that is, again, the concept of genomics. So in order to do this, we have to have significant computational biology support. Take, for example, the BRCA2 gene. So the BRCA2 gene, again, which ultimately will result in a protein, is a very large gene. It's spread over 27 exons. It has 3,000 plus amino acids in the final protein. And again, BRCA2 is a functional protein in the homologous recombination repair pathway. So if you break this down, if there are over 3,000 amino acids, each amino acid is composed of 3 base pairs. So you have over 10,000 base pairs, and if each base pair is basically made up of 2 nucleotides, you have over 20,000 nucleotides that make up the BRCA2 gene. Any change in one of those nucleotides is a variant, is a mutation. And so it's important to understand that even though you have a mutation, it doesn't always necessarily mean that it is going to result in a non-functional protein or something that is going to increase the patient's risk. So again, to put that into perspective, you can have point mutations, you can have a change in one of the nucleotides that alters protein function. So in this particular example, a single change in a base pair may result in one of those stop codons. And remember, that's why I started this talk really highlighting the fact that the body is very, very structured. And if you have an alteration in that sequence that results in a stop codon, it will stop the translation of the protein. And more than likely, that will result in a truncation or a loss of function of that protein, which in this particular case, in many cases, can result in a loss of function and an increased susceptibility to cancer. However, you can have a silent mutation. And in this case, if you look at the top, which is really the normal DNA structure, if you look at the glycine, which is coded for by GGC, if you have an alteration in the C to a uracil, that's still, because of the fact that of the degenerate code, that still results in the creation of a glycine amino acid. So in this particular case, you have a mutation, but there is no change in the amino acid. So therefore, this particular mutation is not going to result in a stoppage of translation. So it's a mutation, but the change in the amino acid sequence doesn't alter the protein formation. In this particular case, the change in the nucleotide results in the formation of a serine amino acid versus in the normal sequence, a glycine. And so the question is, does that alteration in the amino acid sequence result in a change or a loss of function in a protein? So again, when we talk about these proteins and we talk about these mutations, it's critical to understand that not all the time you end up with something like this, where normally you have a lysine, which is coded for by AAG. You have a change in the nucleotide from an alanine into a uracil, or excuse me, an adenine into a uracil, and now you have this stop codon. So it is this stop codon that will then stop the translation, and again, more than likely will end up with a non-functional protein. And again, this is where the problems arise. So as we test more, we have a very standard classification. So just because you have a variant, a mutation, doesn't necessarily always mean that it is going to result in an increased risk. And so remember, you don't inherit cancer. You inherit the risk for cancer. So the standard classification, if a variant is detected, comes under 5 categories. It is pathogenic, so that means the sequence change likely will contribute to disease development or an increased risk of development. You have likely pathogenic. You have a variant of uncertain significance, which generally means that the mutation, the variant, has been identified, but it hasn't been identified enough to be able to say with definition whether or not it increases the risk of the patient. You have likely benign and you have benign. So it is this genetic variant of uncertain significance that really causes a lot of problems. And again, I think it should be really noted that just because you have a VUS doesn't necessarily mean that the patient's at risk. It just hasn't been identified enough. Because these are germline, these are inherited changes, the sequence itself won't change. However, the identification of a particular variant in relation to a disease just hasn't been tested enough and identified to result in any type of certainty. So these VUSs really need to be monitored. And over time, as testing increases, these actually can be reclassified at some point either to something that's pathogenic, likely pathogenic, or on the other hand, it could go benign. So unfortunately, what happens a lot of times is that providers will overtreat these. They will treat VUS like pathogenic variants, which is not always necessarily the wisest thing to do. So when you see a VUS, it's just important to understand that over time, this may be reclassified. So again, it's important to understand that you don't inherit cancer, you inherit the risk for cancer. So this is the Knudsen two-hit model. And so why hereditary cancers are oftentimes detected in a much earlier age is, again, remember, you have inheritance of a copy of each of the genes. So you get an allele from each parent. So if you inherit that, you inherit an infected, right, or a defective allele. So most of the time, the body, the cells can still function and result in a functional protein with the loss of one of the alleles. However, it is when you get the second hit, the loss of the second allele, which again is usually the environmental interactions, the environmental factors, smoking, radiation, those types of things. That is where you then get the loss of protein function and the unregulated cell growth. So that is why hereditary cancers tend to be diagnosed at much earlier age, because you already have an inherited loss of function. When you have sporadic cancers, which again, the majority of prostate cancers are, it takes time to develop the loss of both alleles. So again, all of this has really been trying to understand, to get the urology world, or in that case, any provider that's interested in cancer biology, is to have an understanding of the basics, which we all know, which is central dogma, DNA to RNA to protein, but now we have to tie that knowledge into clinical relevance. So over the past 10 years, we've identified certain genes that have been associated with prostate cancer risk. And many of you have seen these genes. Again, most of them are involved in DNA damage repair. HOXB13 is an AR repressor, androgen receptor repressor. It is probably the most commonly identified in patients with inherited prostate cancer, but it does not differentiate aggressive versus non-aggressive cancers. But the clinical relevance of this is that, again, we know that in patients with metastatic castration-resistant prostate cancer, about 23% will have biallelic DNA repair mutations. And again, this is involving both homologous recombination and mismatch repair. We also know, and again, this is important from a histopathology standpoint, if there are patients that we will see on biopsy reports that have ductal or cribriform histology. In those patients with that type of histopathology, overall, about 49%, close to 50%, had DNA repair gene mutations. The majority of them are in this homologous recombination, but you can also see them with mismatch repair variants. We also know that in patients with higher grade Gleason's, that there is an incidence, there is an association with mismatch repair. And again, it is these mismatch repair genes that are associated with Lynch syndrome and also with microsatellite instability. A seminal paper that was published a few years ago really looked at patients who were candidates for active surveillance, and lo and behold, if they were discovered to have germline mutations in some of these homologous recombination repair genes, mostly ATM and BRCA1 and 2, there was an increased incidence of upgrading at the time of pathology findings. So again, if you are considering a patient for active surveillance, then if they have, again, a germline mutation, they may not be the best candidates because you have upgrading of the biopsy in early stage prostate cancer. So one of the areas that we are keenly interested now, especially in patients with metastatic castration-resistant prostate cancer, is this concept of dual or synthetic lethality. In normal functioning cells, PARP is very important, like I said, in DNA repair. So if you have an alteration or a loss of function of one allele only, these cells can certainly still result in DNA repair, and you have functional homologous recombination and cell survival. However, if you have cells where you are homologous recombination deficient, the inclusion of a PARP inhibitor, which now there are multiple available commercially in the market, you can add that, which results in dual lethality and cell death. So the importance of this is that identification of patients with homologous recombination repair mutations lends itself to the addition of PARP inhibition, and it is a very actionable mutation. Same again with microsatellite instability. And again, microsatellite instability can also be essentially detected with loss of expression of the mismatch repair genes or proteins. So if you have a loss of expression of MLH1, PMS2, MSH2, MSH6, those patients, by definition, may have microsatellite instability and therefore may be candidates for certain immunotherapy or checkpoint inhibitors. So, with all that being said, it is important to understand that these genetic drivers are very important in the management, and we need to start incorporating that into our testing in the evaluation and management of these patients. So again, these are the most recent NCCN guidelines regarding the principles of genetic testing and molecular biomarkers. And again, much of this is related to personal history and family history. And these are the guidelines for somatic tumor testing. And again, these slides will be made available to the audience, and it's very important to go through these because this will give you some guidelines on how to test and who to test. And why this is important, and again, this is data that comes from not prostate cancer patients, but from Illumina, which is a testing company that makes the sequencers, is that even amongst medical oncologists, is that even though we think that medical oncologists may be doing a little bit better than us, is that based upon data is that there's 100 metastatic cancer patients, and this is for lung cancer patients, and 60% underwent no molecular testing, 25% had single gene testing, and only 15% underwent multi-gene panel. So again, for every cancer, not just prostate, and we're understanding more about bladder as well, but certainly those with breast cancer, colorectal cancers, ovarian cancers, et cetera, we are starting to understand that in this day and age of precision medicine, we need to incorporate this into the management of our patients. And again, this is really looking at patients of over 4,000 patients with non-small cell lung cancer, and that only 14% of patients are candidates for targeted therapy in this particular study, 14%. However, in those patients that were actually tested, it resulted in changes of therapy, which resulted in a 63% overall longer survival. So again, the identification, especially when you have actionable genes that results in therapeutic changes to the next line of therapy, can result in longer patient survival and better outcomes. So again, in conclusion, it is important to understand the basics of genetics and evolution of cancer. Gene mutations or variants are not always associated with increased risk, and they also need to be always discussed in the context of a specific disease state. And what I mean by that is that we may be identifying genes or variants, but it needs to be recognized that we are testing, in our case, in relation to prostate cancer. How that variant, how that gene is relative to other disease states, both cancerous and non-cancerous, really cannot be addressed as again, all of these disease states have certain genes that we know put the patient at increased risk. Current technology, next generation sequencing, really is outpacing our current understanding of its clinical relevance, but it's important that as we start to test more, we will have a better understanding of these molecular drivers, and we will need to operationalize genomic testing in our practices. So this is one of my favorite quotes, is that knowledge that does not change behavior is useless. So as we incorporate this, we need to understand its relevance and, more importantly, on its potential on how we can better change our current practice patterns, our therapeutic options to result in optimal patient outcome. Thanks so much. Again, these slides will be available to the audience. And if you have any problems or questions, below is my email, and I will be happy to address anything that comes my way. Have a good day.